Welcome to the light reactions. This is part of the metabolic processes guide, and the light reactions are the first part of photosynthesis. And in the light reactions, we look at trapping energy from light and converting it into something useful in the plants. In the light reactions, we basically start with light, a coenzyme known as NADP+, water, and ADP, an inorganic phosphate. And through the series of reactions along the thylakoid membrane, you will get an output of a net increase of ATP, reduced form of the coenzyme NADPH, and you will have a byproduct of oxygen. So the key here is when you're working with the understanding that's going on here, you want to make sure that you remember the pigments. Okay, so we look at pigments, and often with pigments, in green plants you have chlorophyll A, chlorophyll B, xanthophyll is another one, and your carotenoids. Okay, so when you're looking at those, remember this is your green, yellow, and orange. So those make up the light centers within the photosystems. And when we get to the next slide, hopefully that will make sense. And you have to remember, in talking about metabolics, quite often they are redox reactions, which is the oxidation and reduction. And the thing I like to remember is Leo the lion says grr. And if you're looking at that, that's loss of electrons is oxidation, gain of electrons is reduction. I know that sort of seems like a misnomer, but that's the way the chemists have started it, and that's how we need to remember. And also remember that a hydrogen atom those hydrogen atoms, when we look at it as an H, is actually a proton, or an H+, and an electron. So quite often in oxidation and reduction in biological and metabolic processes, when we talk about oxidation, we're actually losing an entire hydrogen. And when you're reducing, you're actually gaining a hydrogen. So try and remember that when you're looking forward at the next couple of slides, okay? So let's take a look at the thylakoid membrane. And the thylakoid membrane is made up of a whole bunch of different proteins and coenzymes within it. So I'm just going to quickly label them. This first one is the photosystem 1. The last one over here is photosystem 2. Again, this is where you find your pigments. This in the middle of that triangle right there, that's your cytochrome. And then there's the little bits in between. This over here is NADP reductase. And if you notice, inside here, this is inside the thylakoid, so this is the inner thylakoid space, and out on the outside is your stroma. Okay, so please remember, when you're looking at this, you have to have your whole picture of what you're looking at. Because if you forget, then it sort of loses a place. Over here, what you have is a proton channel. And at the end of the proton channel is your ATP synthase. an ATP synthase basically is, when you look at, remember when you're looking at enzymes, A's, enzyme, synthesis, so you're putting together ATP. If you look up at NADP reductase, again, enzyme, and reducing NADP. So you're going to reduce NADP at that enzyme there. You're going to make ATP over at this enzyme. And then you've got your other different coenzymes. So I'm not going to worry about it, but this right here, I will highlight in blue, if you can see that. That blue, this blue are just electron carriers. One is ferredoxin, and plastoquinone and plastocyanin. For those that are really wanting to learn that stuff, you can, but for this class, you don't need to actually remember it all. Okay? So let's continue on, and I'm going to go to the next slide without it all labeled. So if you can remember, and you go through these, can you remember that photosystem 1 is here, photosystem 2 is here, the cytochrome is in the middle, NADP reductase is over here, and then the ATP synthase is that round at the bottom. Okay, I'm going to flip it around and it's going to be on one long chain. So if you go to slide and you see this, the outer bit here is the stroma, and the other side on this side would be the inner thylakoid space. Okay, and so we're going to start, different books look at different things, so I'm going to start at photosystem 1 because that's just sort of how I like to think of things, and it makes more sense to me. So the sun is shining nice and bright, and if it hits photosystem 1 with the right wavelengths and the pigments are energized properly, it's going to excite an electron here. And that electron gets excited and it will get pulled from photosystem 1 over to the NADP reductase. Okay, so you're going to take the electron from photosystem 1, it's all excited, it's jumped out of its electron shell, and it gets grabbed by ferredoxin and pulled over to the NADP reductase. Sitting outside waiting for it is NADP+. So NADP+ will come in, grab the electron, and it will also have a proton there. I'm going to put the proton in red, and hopefully I can remember that it's going to be red. It enters there as well, and then we end up getting a compound of reduced NADPH. Okay, so once the electron has left photosystem 1 and goes to photosystem 2, NADP+ picks it up and becomes NADPH. And we've taken that energized electron and we're going to take that and we're going to use it further on in the Calvin cycle. That nice energized electron will be converted into more of stored energy later. So now that we've lost the electron here, okay, there's an electron missing because it moved over, we need to replace it. So photosystem 2 does the honors. Photosystem 2 gets excited. The electron gets excited in photosystem 2. It gets pulled over to the cytochrome. And we'll talk about and at the cytochrome, it uses that energy to grab a proton that's floating around outside, and I knew I'd forget that, so we're going to take the H. That negative and positive are going to be attracted. The H comes closer to the electron, and the electron pulls it all the way through and puts it inside the inner thylakoid space. And if you notice, if we start pulling H's on one side, we start developing a gradient of protons on the one side. And then that electron will continue on and replace at photosystem 1, and photosystem 1 excites it and keeps moving on. Okay? So once we've lost the electron at photosystem 2, okay, we've lost that electron, so it's now oxidized, we need to find a source of electrons. And this is where what we have called and it sort of sits right next door to photosystem 2. That's photolysis or photolysis, depending on your emphasis or emphasis. And photolysis, basically, you have 2 water molecules, okay? And you have 2 photons of light striking it. And it's going to lysis, cut, using light. You're going to break apart water, and what you end up getting are 4 electrons from the hydrogen, 4 protons, and an oxygen molecule. Okay? And so in photolysis, what you've done is you have now created a bank of electrons, so those 4 electrons can now be fed up into photosystem 2. The 4 protons are now here creating more of a gradient on this side, on the inner side of the membrane. So you have what's called a chemiosmotic gradient. Okay, so now you've got this chemiosmotic gradient. You've got the electrons being fed through, forming NADPH. Now, remember there's a proton channel here. Knowing that this is a cell membrane, and we learned about membranes being impermeable or selectively permeable, but they were impermeable to ions, so protons can't naturally flow through. So what they do is if we open up a channel, those electrons are going to start flooding through and going through. And as they go through, this is ATP synthase, and what happens at the active site there is ADP mixing with adenosine, like adenosine diphosphate and a phosphate produces ATP. And because it's on the active site in here, it can produce the ATP no problem, but there's not enough energy after the reaction to take it off the active site. So what happens is when a proton goes sailing by, it just pushes the ATP out, and now you have free ATP, which can now head off to the light reactions. So if we were just to summarize it up, you've got oxidation taking place at photosystem 1, which then takes it over to NADP+, and then you reduce. You have oxidation taking place at photosystem 2, shifting over to photosystem 1. And it continues onwards and upwards, and then you have to have water as a source that you can break apart to create electrons, protons for the chemiosmotic gradient, and oxygen, which is now your byproduct, and it is now Willdane waste. So that's basically the light reactions in a nutshell. So if you have other questions, please make sure you contact me. Otherwise, hopefully this makes light reactions a little clearer.